My contrast is the powerhouse of the sun. Oh my god! I have a bio test today. I have a chem test this week. I'm sad. I gotta go to school. I gotta learn. Get those co-ops. Get those internships. I gotta keep my GPA. I gotta get a job. I gotta. The complex system of signal pathways in the brain is built upon the process of neurogenesis. A nerve cell can develop into one of three roles in the nervous system. It can become a glial cell, a neurocell, or it can never develop. The neurocell is the foundation for all cognitive and reflexive actions by its characteristic synapse, where neurotransmitters and other hormones are transmitted from the axon of one cell to the dendrite of the other. Glucocorticoids are one of such signals which activate the parasympathetic nervous system in response to stress. The role of neurogenesis is that it creates multiple synapses, creating alternative pathways for cells which act as a buffer in minimizing the effects of stress. If stress continues, though, neurogenesis can actually degrade. In a study comparing mice with and without neurogenesis, it was found that when exposed to stress, mice lacking neurogenesis had increased food avoidance, increased failure in the swim test, a decrease in sucrose preference, and overall increases in anxiety and depressive behaviors. The major conclusion drawn from this is that as neurogenesis decreases, neutralization of stress also does.
A 2006 study explored the link between the elevation of glucocorticoid levels in the brain, lack of sleep, and reduced neurogenesis. It used the small platform method of sleep deprivation to keep mice from obtaining REM sleep. Mice were placed on small, suspended platforms to prevent them from falling asleep, while platforms large enough to cause no disruption were used as controls for factors such as stress. To measure neurogenesis, they used a method in which bromodioxyuridine, or BRDU, a thymine analog which can be incorporated into newly synthesized DNA, is injected into the mice, as described in Toppin 2007. The number of brain cells incorporating BRDU can then be used to track cell proliferation. Greater numbers mean greater cell proliferation and therefore greater